Hello, everybody. Welcome to the Atto Fridays. It is really a pleasure to have here today a very distinguished speaker, Professor Gerhard Paulus. In fact, I was just talking to him and I heard that he is also a fellow YouTuber, that he has a YouTube channel with a lot of followers. He is the chair of nonlinear optics at the Friedrich Schiller University, Jena, Germany. He graduated in 1992 from the Ludwig Maximilian University in Munich, Germany, where he also completed his PhD in 1995 under the supervision of Professor Herbert Walter. And from 1995 to 2003, he was a research associate at the Max Planck Institute of Quantum Optics in Germany, where he also did his habilitation in 2002. And from 2003 to 2007, he was an associate professor at Texas A&M University. And since 2007, he's a full professor at the Friedrich Schiller University in Jena, Germany. He was also the head of the Atomic Physics Division of the German Physical Society from 2018 to 2020, and the Dean of the Faculty of Physics and Astronomy of the Friedrich Schiller University. And he won also the Best Practice Award competition, Mint Digital of the Stifterverband, and several other teaching awards from the University and Student Council. And I have to say as well that, as I just announced, I just found out he's a fellow YouTuber. He has a YouTube channel with his lectures, and he has also awards for that. He's a Batsheva, the Rothschild Fellow of the Israel Academy of Sciences and Humanities, and has a honorary doctorate from the University of Sofia in Bulgaria. And his contributions to strong field and attosecond science include discovery, proposal, and the correct explanation of the above threshold ionization plateau in the 1990s. In fact, this year, the plateau has become 30. And he worked also on photoionization with few cycle laser pulses, non-invasive cross-sectional imaging with nanoscale resolution, and the combination of X-ray spectroscopy with high-definition polarimetry in a novel experimental technique that gives access to the electronic structure of condensed matter. And finally, for several years, this is something beyond science, but it's very important, especially as the world is getting crazy. He felt compelled to stand against right-wing extremism by organizing the Klang der Stolpersteine. And these are concerts at all the Stolpersteine in Jena, commemorating victims of the Nazi terror. And the team organizing these last year was awarded the Jena Prize for Civil Courage. So welcome, and thank you for joining the Atto Fridays. We really appreciate it. And today he will talk about XUV coherence tomography, non-invasive, nanoscale, cross-sectional imaging with high harmonics. So the floor is yours. We have also a very fancy background, as you could see. And welcome. We are ready to start.
Yeah, thank you very much. Thank you for the kind invitation and for the very long introduction, basically telling everything what I did so far. Thank you very much. Okay, XUV coherence tomography. What has it to do with atto science? Let us see. So, discovery of high harmonic generation. We use actually high harmonics, and you may have seen the recording of Anne's first interview after the Nobel Prize for her was announced. Remember the scene where she just dismissed her class and learned about the news? In this interview, she made the point that the high harmonics have a significance far beyond attosecond science. I've also made this point often, but, well, if people don't really listen to Anne, you can't expect that they would listen to me. But maybe I can convince you today. However, as a backup in some sense, I'll provide also the attosecond perspective of this imaging method that I will explain. So, and this is called XUV coherence tomography. and I'm pretty sure that you will learn at least one point that's actually useful for your life. Yeah, more or less useful. Okay, so I'll come back to that. Don't want to promise too much. So here's actually a photo that was taken at the recent XRM conference. So in the there you see Anne Lullier, and she is actually kind enough to explain what I'm going to explain today at that conference. My students were sitting in the audience and sent me the photo while Anne was speaking. Okay, yeah. So high harmonics. You see this this famous figure with the discovery of high harmonic generation, published in the famous Journal of Physics B rather than in Nature. What you don't see are units at the ordinate, right? So it says relative intensity of harmonics. So the absolute flux is low. Now, one can always argue whether the glass is half empty or half full. I prefer a positive view on the world. I would claim the flux is high, much higher than one could have dreamed of. People say the efficiency is low, 10 to the minus 4, 10 to the minus 8. Well, on the other hand, I never heard people complain about the efficiency of synchrotrons. So as a homework problem, try to find it out. No, I don't say the solution. So nevertheless, efficiency is a challenge, sure. On the other hand, today's lasers are different from those used by Anne at that time in 1988. Here you see a more recent measurement. And what you see in particular are that there are now units here. So photons per second and electron volts. And there's a number, 10 to the 15. So these are numbers that were previously only known from synchrotrons. Of course, synchrotrons will win the harder the photons. Nevertheless, high harmonics still have their unique selling points. I think I don't need to convince this audience. Well, just to illustrate my point, as a side project to coherence tomography, we measured the broadband reflection of an aluminum 203 substrate. So this is corund. Same thing that you would use basically in a titanium sapphire laser, right? And what you see there, so just below 80 electron volts is a prominent Nexaf structure. And usually you would use a synchrotron in order to measure that. And what you see is the reference. This is actually the red line, right? And blue is our measurement. And you see virtually perfect agreement. And the point is, we measured that much faster because we used a broadband technique. So we measured basically all wavelengths at the same time. And at modern synchrotrons, you have to tune usually the wavelengths with the undulator. Yeah. Okay. So you see, high harmonics are actually useful beyond attosecond science, even at this small example. High harmonics. I said synchrotron-like photon flux, at least for photon energies below 100 electron volts. It's diffraction limited. Only the newest generation of synchrotrons can compete with that. And it's ultrafast. Yeah, so you would need a free electron laser, and they are really expensive. You would need a free electron laser in order to be able to compete with the temporal resolution of high harmonics. Okay, using high harmonics for something different than attosecond pulse generation. So namely imaging. So this was one of the things that Anne Lullier referred to. And the most well-known and most widely used technique for imaging with synchrotrons, but also with high harmonics is the so-called Tomography technique. It was invented by Walter Hoppe in the framework of electron microscopy, actually, right? And the idea is to raster scan the sample like you see here, so to raster the incident radiation at different positions and to record the diffraction pattern, and then calculate from the redundancy of the information that you gain here, then to reconstruct the phase that is initially thought to be lost. It's not entirely lost. It can be reconstructed. Tomography. So Walter Hoppe was, of course, born well too early. One needs powerful computers in order to do that. Powerful computers like everybody has on one's desk nowadays. But, you know, in 1986, so this idea came a little bit too early. Okay, good. Tomography, strange name, right? What does it mean? So it means folding. It's, of course, Greek. So obviously, Walter Hoppe had a humanistic education. And the term is perhaps known to some of you from this kind of paintings, where you can fold this painting, right? So, and this is called a triptychon because there are 3 panels that can be folded into each other. Yeah, by the way, this is a painting of Lucas Cranach the Older. And what you see here are the dukes of my region. Yeah, so this guy here, he founded the University of Jena. So he's called, his name was John Friedrich the Magnanimous. And these are his ancestors. Yeah, so John the Steadfast and here Frederick the Wise. I have to say and admit, yeah, so that the founder of my university, that he was not particularly wise, I would say. He lost a stupid war against the emperor and therefore lost most of his dukedom, including the university in Wittenberg. And so he had to found a new one. And this was Jena. So this is why I'm here. Okay, at least the consequences were not so bad, at least not for me. Good, tomography. Here's an example, actually 2 examples. This is a Siemens star, and you see the resolution, a picture made by colleagues in another group here. And here they looked actually at a semiconductor device and were able to reconstruct all the structures and even to a certain extent also the different materials that are used in this semiconductor device. The point is, tomography is mostly a lateral imaging method, like most imaging methods. What I want to talk today about is cross-sectional imaging. So we want to look into the sample. Yeah? And how this goes, I will explain, of course. So tomography, simple and robust, no lenses. Yeah, so this is the decisive thing and the decisive problem in XUV, that so the region is also sometimes called vacuum UV because basically all materials absorb very strongly, right? And therefore you can't use lenses. Yeah, robust imaging reconstruction and provides phase information. It needs monochromatic radiation. This is not so nice about tomography. You know, it's pretty expensive to produce these high harmonics, right? And then if you have to throw away all harmonics but one, that's painful. Yeah, and of course it also has no or just limited depth resolution. So optical coherence tomography, or then XUV coherence tomography, is the solution that we propose. Yeah, so optical coherence tomography is coherence tomography in the visible, and I'm going to introduce that one. It is actually known from the ophthalmologist's office. Yeah, so optical coherence tomography was invented 35 years ago or so, and now it's a business of billions. So this is what you find in a modern ophthalmologist's office. At some point, I went to the ophthalmologist's office here in Jena. I didn't know the doctor, and vice versa. He asked what was wrong, and I replied, I need an OCT. Yeah, of course, he was surprised and puzzled. And so I added that I would like to have a coherence tomogram of my retina in order to show off. To show off in talks like this one. This is my retina. Yeah, so you see the fovea here and the typical layer structure that you have in the retina. The doctor was quite satisfied with that, yeah, so don't be worried. So the question is, how does one get this? Yeah, so OCT is really remarkable. They have a resolution. Yeah, so at a recent visit, he told me that his newest device has a resolution of 2 micrometers. Can see basically every cell. Yeah, so really, at least in principle, really remarkable. Okay, so how does it work? The simplest way to explain it is actually with a Michelson interferometer and assuming short pulses. You don't need short pulses, you only need short coherence lengths. So this one here is the femtosecond laser that produces the short pulses. Once again, we only need a broad bandwidth, short coherence lengths, right? But it's very easy and intuitive to understand if we assume pulses. So we have a beam splitter, right? And just to make the different paths distinguishable, I gave them different colors. Of course, the color remains the same in this mirror, right? So here's the sample, and it has a layered structure. And therefore, at each interface, a replica of the pulse will be reflected. So back will come 3 replicas of that pulse. And the other half here went to the reference mirror and also comes back to the beam splitter. And then they are recombined. And what you see here is that they don't overlap, and therefore there's no interference. However, we can, of course, move that reference mirror. And if we do that, yeah, then, of course, we can overlap these pulses and we would record an interference pattern disappearing and appearing according to the depth structure of the sample. This is how OCT works in principle. This is how this picture of my retina was produced in principle. In reality, it's a little bit different. Yeah? So one doesn't want to use moving parts and stuff like that. Therefore, one rather does this in the frequency domain. Yeah? So instead of moving the mirror here, people record the spectral interference of all these pulses. Yeah? So basically, the grading makes these pulses long, and then they overlap, and they interfere here. And you would then have such a spectrum showing all the spectral interferences. And of course, this is just the frequency domain of what was previously been the time domain. So you can use a Fourier transform in order to go back and forth between both. Yeah. The Wiener-Khinchin theorem does the transformation. Okay, so now let's look at a few. Let's look at the scaling. Yeah, so what I have here is the formula for the coherence lengths. Yeah, and the coherence length depends on the bandwidth of the radiation inversely. Yeah, so the larger the bandwidth, the shorter the coherence time, right? But it also depends here on the wavelengths, actually on the wavelength squared, right? And now if you think about doing this in the optical domain and in the XUV, in particular doing it with high harmonics, right? So high harmonics, you have a huge bandwidth, and you have short wavelengths. Yeah? So I said that the OCT apparatus of my ophthalmologist has 2 micrometers of resolution, will have 100 to 1,000 times higher resolution in the XUV. Okay, well, this is actually mentioned here. And of course, we don't need to throw away all the expensive XUV photons. Yeah? So we make efficient use of the harmonics. So I said something about the bandwidth. Yeah, now I have to go back to what I said earlier, namely that materials absorb very strongly. And when I had first this idea of using high harmonics for doing coherence tomography, then this was when I was still in Texas, actually. My second thought was that this is an incredibly stupid idea. Well, because the spectral region is called vacuum UV. But there are exceptions. And silicon is a particular nice one because silicon has a transmission window between 30 and 100 electron volts. Yeah, I don't need to say anything about the importance of silicon. And also, I also don't need to mention that one can create high harmonics particularly nicely in this region. But anyway, the coherence length is now no longer, or at least not only determined by the high harmonic spectrum, it's also determined here by this transmission window, right? And if we take this transmission window and calculate the corresponding coherence lengths, then we'll find something like 11 nanometers. That's the resolution. Yeah, so that's the limit one can achieve for silicon. Another prominent example is, of course, the border window. I know it's much harder to produce harmonics, high harmonics in this region, but of course, what I'm going to propose can in principle also be done at a synchrotron. Yeah. So, and then if you look at the transmission window of order, then you find actually 3 nanometers of depth resolution. They're really interesting quantities. Okay, now that we discussed this, let's go to XUV coherence tomography. The question is how to implement it. Basically having no mirrors. Yeah, so mirrors are not very highly reflective in the XUV, also the sample not. Yeah. And even more difficult are beam splitters. Yeah, so they are practically impossible to realize, because you would need to have a flat, ultra-thin surface of a certain extent. Yeah, so that's difficult, at least. Okay, so the idea is to make it simpler, and actually we made it very simple. Yeah, so basically we use the sample as the beam splitter. So we take as the reference beam the reflection at the surface, right? So this reflection here at the surface. And we record the spectral interference of this reflection at the surface with all these reflections, with all these internal reflections. And in the lab, it looks like that. Yeah? So high harmonic generation is the smallest part, right? Then one has to get rid of the fundamental. Yeah? So the efficiency is only, well, 10 to the -5 or something like that in our case. Yeah, so you need to get rid of several watts on the background. Yeah, so you have several watts on the background of microwatts, if not nanowatts of the XUV radiation. Yeah, and then there's a focusing unit that focuses the XUV radiation on a sample, and the sample is situated on a sophisticated nano positioning stage. You'll see in a minute why this is so sophisticated? Well, for the reason I'm going to tell, but also for other applications that we might have. And what we do is that we record the reflected spectrum, right? This is done in a high resolution XUV spectrometer that my co-workers developed here in my group. And actually, they founded a company and sell this device and other devices. So if you're interested, go to their website. Indigo is the name of that company. I have no financial interest in the company, right? So, okay. Well, one problem. Today there aren't problems, aren't there? So we speak of challenges only. Yeah? So the challenges are the fluctuations of the harmonics. And high harmonic generation is, of course, a highly nonlinear process, thus amplifying fluctuations of the driving laser. And our femtosecond laser is already fairly old, more than 15 years actually, but there's a cure. Yeah? So what you see here is a thin plate, a nanometer thin plate, that we position with all the sophisticated mechanics, nanomechanics, just in front of the sample in order to get a live reference. Yeah, so we record on the camera actually two spectra. So one that was reflected at the surface at this thin foil in front of the sample, and one that went through the sample, yeah, back and forth, and made it then to the spectrometer. So you see that we have here two spectra. One is the reference spectrum, and the other is the spectrum modulated from the spectral interferences due to the internal reflections in the sample, right? And with this, we can kind of, well, normalize and get rid of all the fluctuations that we have in the high harmonics. Of course, it would be nice to have a better laser, then we would have even better results, but well, that's always the case. So this is how you do that. So I should mention that here. So just to show the difference. So this is without this live reference, and this is how it looks with live reference. So a drastic improvement. Yeah, so and the advantages are mentioned here once again. Now, XCT in action. So what you see here is a coherence tomogram of a sample. It's a silicon sample, and we prepared it such that two gold layers are buried in silicon at a depth of 200 and 300 nanometers. Yeah, so you see here these two gold layers, and we even put some letters on it, XCT by chance. Yeah? You also notice that there's more actually. Yeah? So you see this layer here, right? So this layer here, and this is actually a ghost. Yeah, so we have to deal with that. It comes from interference between the two gold layers, right? So what we would like to have is interference between the surface and the first gold layer, the surface and the second. But we also see a signature here from interference between these two layers. So this ghost layer is here at a depth that corresponds to the distance of these two gold layers. Okay, so this is understood, this layer here, right? We will have to deal with it. We'll have to get rid of it. But there's another one. Yeah? So if you look carefully, then you see that there is a faint layer, additional layer. It took us a while to find out or to have a suspicion. We asked our colleagues who produced this sample, and they told us finally that at some point during the production, they inspected the sample, so broke the vacuum. And so they said there might be a 2 nanometer thin layer of oxide, but you won't never be able to see it. Well, actually, it's a trademark of optical coherence tomography and therefore also XUV coherence tomography. It's a trademark that you have a very high sensitivity, a very high dynamic range. Yeah. Yeah, here is, yeah, so we had to prove that, of course. And so we cut the sample in pieces. So we took a FIP and cut out a thin slice, a very thin slice, and investigated it under the electron microscope. And sure enough, you easily see here the gold layers. But if you look carefully, you also see here the oxide layer, right? And it was exactly at the right depths that was found also by XCT. Good. Well, now this artifact, this ghost layer, we had different ideas on how to remove it, but finally the best idea was to do phase reconstruction. So initially we thought it won't be possible because it's a one-dimensional problem, but if you do it right, then it can be done, and you see the result here. So the ghost layer has disappeared, and the oxide layer is, of course, still there. All right. Very nice. Yeah. So we know the reflected field. Yeah. So if you know amplitude and phase, then you know the reflected field. And if we know the reflected field, then we know the reflected attosecond pulse train. So think of it like that. I'll make the point later again. The high harmonics are attosecond pulses. So if one attosecond pulse arrives, like the animation that I've shown, then at the sample, at each interface, a replica of that attosecond pulse will be reflected. It won't be an exact replica, of course. It will be a distorted replica according to the spectral reflectivity and the dispersion and so on and so forth. But it will be a train of attosecond pulses that's reflected by the sample. So attosecond pulse trains. A colleague made an animation. Yeah, so it still can be improved, I admit, but nevertheless, yeah, so here you have the incident XUV attosecond pulse here on the sample. You see this layered sample, right? And at each interface, there's a replica. They look all the same here. They should be a little bit different. And of course, after the grading, they should be longer. And in the detector, you would measure the spectral interference. Attosecond pulse trains. So we measure the spectrum. Yeah, we reconstruct the phase. And this means that we now know the reflected field. We know the attosecond pulse train that comes back from the sample, right? So like it is shown here, so we have this pulse here that's incident on the sample, right? So you see it has a few interfaces here, say 20 nanometers thick, which is approximately the resolution that we have. And what could come back would be two attosecond pulses separated by 130 attoseconds. Yeah, so if we know the phase, if we have reconstructed the phase, then we can reconstruct, or then we can do the Fourier transformation, and we know the train of attosecond pulses. And this we can use in order to learn something about the interfaces, about the material more generally. So that's the interesting thing here. So what we do, for example, is that we single out, that we window out one of these pulses, Fourier transform it back. Yeah, then we have the reflection only at that interface. Right? We can shift the frequency back to zero frequency, and then we have the spectral reflectivity, the spectral field reflectivity, to be precise. Yes? So very interesting that one can do that with well, with a fairly simple setup using pulse reconstruction algorithms and high harmonics, of course. Okay, so here's a more complex sample. So what you see here is that the top layer is silicon oxide. Then we have silicon. Then we have a thicker layer of silicon dioxide. Then again silicon, titanium, silicon, titanium oxide. Question is, can we reconstruct such a sample? Of course we can. And what you see here are the reconstructed reflectivities, spectral reflectivities of the individual layers. Yeah, so in particular silicon dioxide layers and titanium and titanium oxide layers, right? And the red curve is the experimental line, and the blue one, the data that you can find, for example, in the Henke tables. Right, so you see slight differences. We think that it's mostly laser fluctuations and spectrometer alignment that is responsible for these residual differences. Meanwhile, we have much better examples. Also, I'll show this later. Okay, so which parameters can be obtained by comparing experiment and model? Of course, we can identify the material because of absorption edges, yeah, so which are characteristic for material. We can, of course, reconstruct the layer thickness. Actually, once we know how the sample is composed in principle, yeah, so once we have a model of the sample, we can do this with a much higher precision than the nominal resolution. Yeah, so nominal resolution is here around 20 nanometers, but actually it's possible to register changes, for example, in the thickness of less than an atomic layer. Yeah, so it's really remarkable. Yeah, and one can also access the roughness, and yeah, so the rougher the material, the less reflectivity it would have. Okay, so this is actually the example from the material sciences I referred to. In that case, it was, as a material scientists, our colleagues from that department, they are interested in alloys. And here, this is an aluminum copper alloy, and here a structure containing of this alloy and aluminum. In between, this we didn't know initially. Of course, for material scientists, it's obvious. They are oxide layers, very thin oxide layers, right? And when we first looked at the spectrum and tried to reconstruct the phase reconstruction algorithm failed. And so we asked them what could be the reason, and they said, of course, you have to, there's an oxide layer, and once we included that oxide layer, it converged immediately. Right, and this is the reconstruction here of the spectral reflectivity, and the dotted curves are the error margins. Yeah, so it's really precise, and one can compare it, of course, to yeah, so these are the numbers for XCT. This for the transmission electron microscope, and here atomic force microscopy. There you find the biggest differences here for the roughness. But of course, the roughness here had to be measured before the sample was completed. Yeah? So it was done during the production, before the final layer was deposited. So one can't really rely on that number. And well, similar arguments for the cases where there's a deviation. So the agreement with established imaging methodology is actually very high. You could ask, what's the advantage of XCT as compared to electron microscopy? Well, one is obvious. For electron microscopy, you need to cut the sample. Actually, you need to do more. You need to prepare it. You have all kinds of, so from the cutting process, you introduce typically gallium. And for this kind of sample, there's a special problem that was also a surprise to us. Electron microscopy puts much more energy into the sample than we do. And for such a sample, this is particularly problematic because it might melt, right? And then the sample is gone. So there are specific and significant advantages of imaging with using XUV. Yeah. Okay, this is here shown once again. Yeah, so this idea of attosecond pulse, of interference of attosecond pulses. So this was attosecond pulse strains. Now a few more examples, namely 2D materials. Was also a series of works that we did. So in the first example, we just buried graphene into silicon. Yeah, so a monolayer of graphene, and the question was, can we see it? Of course, we could see it. Then we took 2 layers, B layers and 3 layers, and we can distinguish actually whether it's a B layer or a 3 layer. Yeah, so the case of the 3 layer was kind of initially puzzling, and then after you have solved it, then it's of course interesting. So we see here a reduced reflectivity, right? It turned out that this is due to an increased roughness. And this increased roughness, this comes from the fact that if you stack several graphene layers, they tend to wrinkle somehow. Of course, for the people working in that field, that's known since a long time. For us, of course, again, it was new. So we learned it the hard way, as so many other things. Other encapsulated 2D material. Actually, my student Felix Wiesner, who just finished, it's already a few months ago, his PhD. He did a systematic study. Looking at the available data, what kind of XCT contrast we can expect for different encapsulated dechalcogenides. A difficult word still for me. So you have an entire series here, and you can embed them in different material, HBN, and you name it, carbon, and so on and so forth. And you see that in most cases, one gets a decent contrast and reflectivity. So 4% is typically enough. So 4% of contrast would be enough for us. So we tried it out. So this here is the first example. Molybdenum disulfide buried in what was it? I don't remember. Sorry for that. And here's another example. So I think it's buried in HBN in this case. And this then was what people in that field call a device. So it's a structure made of, in this case, so let me explain it. So the gray thing here is a flake of HBN. And the pink structure is also HBN, but it lies on top of the bottom flake. So like a blanket would cover you if you are in your bed. Yeah, so one would still see the outline of your body, right? And sandwiched in between is this 2D material, right? And then we tried to reconstruct this device. Yeah, so has no functionality, there are no wires to it, but nevertheless they call it a device. Yeah, so here's the optical microscope image, and we used different modalities in order to image it. So this was XCT, right? And we reconstructed all the different layers and also verified it again with atomic force microscopy, as you see here, right? And numbers again match very well. Okay, so I come to the conclusion. I think that high harmonic generation is an ideal source for non-invasive cross-sectional and nanoscale imaging. It has, as is typical for coherence tomography, it's a white light interferometric method. It has a very high sensitivity, has material sensitivity. We also demonstrated NEXAFs, has little to do with imaging. And we demonstrated that it's applicable for encapsulated 2D material. The intriguing thing we didn't, well, we have some preliminary data, but the intriguing thing is, of course, the inherent potential for ultrafast imaging. Yeah, so what I dream of is kind of, say, a transistor, and you look into the transistor and watch the time-dependent current. Yeah, so we did the calculations there. Yeah, so for usual current densities, as far as we know so far, it doesn't seem to be feasible. But that's a dream, right? To record ultra-fast processes with nanoscale resolution inside a sample, and of course without destroying it. Yeah, with this, I would like to thank you for your attention.
So thank you so much for this really interesting talk. Beautiful animations as well. And we have here someone with a raised hand, Luli. So please feel free to ask your question. We have actually 3 Lulis, so I don't know who has the raised hand. I don't know. Hello? You can unmute yourself. Okay. No. So we have Antonius Pelekanidis. He would like to know, how do you measure the phase of the reflected beam?
It is reconstructed. Yeah, so it's an algorithm that reconstructs the phase. So what one typically, it's an iterative algorithm. Yeah, so what you have to, yeah, so the most well-known example is actually coherent diffraction imaging. Yeah, so there you have there you have the diffraction pattern, right? And then you just guess the phase, and then you would make the Fourier transformation, right? And you need to have some prior knowledge about the object. Yeah, for coherent diffraction imaging, it's a simplified version of tachography, particularly easy. You have to have what they call a compact support, yeah, which just means that you need to know that your object has just a finite size, yeah? And that the space that you illuminate is larger. And with this prior knowledge, after you transform the diffraction pattern back to real space, yeah, then in general you don't get the object. Yeah? You get something completely weird, but you know that everything that's outside the size of the object, this is the prior knowledge that you put in, you have to blank out, right? So you do that. You make the Fourier transformation, right? You get phase and intensity. And then you would replace the intensity that you calculated, you would replace the intensity by the measured intensity. And then you would do this circle so long until you get convergence, right? And in our case, it's more difficult. Yeah, so perhaps too difficult to be explained here. But in principle, this is the idea. Yeah? It's an iterative phase reconstruction. It's an algorithmic method in order to do phase reconstruction. By the way, some people use similar ideas in order to reconstruct kind of the wave function that you create in photoionization or something like that. Yeah, so then you would know the time dependence of the electron wave packet that goes out. Yeah, so similar ideas.
So Luli, yeah, so now you are around. We were looking for you. So you would like to ask a question, right? I don't know what is happening. Hello? Okay. You are unmuted. You can speak if you want. I don't know.
Oh, yeah. Can you hear me?
Yes. Yes, hello.
Okay. Actually, this field is quite new for me. So as my field is like working the medical image analysis like a 2D or 3D using the deep learning approaches or AI approach. Now I want to integrate the XUV with the AI. So I have the question like the basic start for this ones. Like I already put the questions, and maybe you can find in the chat room.
Yes. So how could XCT be applied in non-invasive biomedical imaging at the cellular level? And what role does AI play in enhancing its capabilities? So thank you. I know we were waiting quite a lot during the talk to ask your questions. So
yeah, so biomedical imaging. So one would have, so of course, I'm not a specialist in biology, right? So there might be other opportunities that I'm not aware of. But usually people would say that you need to go to the water window. For the water window, the problem with high harmonics is that we have not enough flux there. You could still do this at a synchrotron, right? Of course, also there would be problems or rather challenges, namely that the reflectivity goes down with increasing photon energy. But in principle, it's possible. So we did this at a synchrotron, and one can do XCT in the water window, and in principle, one can look at biological samples. So there's one thing that might be a problem there. So I didn't speak here about lateral resolution. Yeah, so the coherence tomogram that I've shown, so the scales for the lateral resolution and for the axial, so the resolution in beam direction is different. Yeah, so actually 1,000 times worse. So what one has to do in that case would be to combine, say, ptychography and XCT. And this is actually something that we are currently working on. It's a pretty tough thing, but we hope that we...
No, thank you so much. Thank you very much.
I'm muted, sorry. Thank you. We also have a comment or question, not so sure, from Tobias Witting. I think the actual spectral phase of the attosecond pulse cannot be measured with this method. The measurement is linear spectral interferometry, which allows to retrieve the phase difference between reflected and incident pulse. So my question to the speaker is, do you agree or do you disagree?
So the claim is that we can't measure the phase.
Yes, of the attosecond pulses.
Well, we did.
You see? So that's always,
Yeah.
you know, when you're giving talks and people comment.
So we measure the so one thing, we know the attosecond pulses if we assume that the incident pulse, so if we know the incident attosecond pulse, then we also know the reflected attosecond pulse, right? For us, the attosecond structure is not important, therefore we don't measure the incident attosecond pulse. But if we would know the incident attosecond pulse, then we also know the reflected pulses and all their phase properties, amplitude, distribution, and so on and so forth. Right? I would say one can be pretty positive in the year 2024 that the incident pulses are indeed attosecond pulses. So I would not be concerned too much about that.
So is this fine with you? You can ask your question as well. Feel free to unmute yourself, Tobias, if you'd like to make a comment.
Can you hear me?
Yes, we can.
Oh yeah. Thanks for the answer. So I guess it kind of so basically, if you know the incident pulse and then knowing the phase difference between reflected and incident pulse, of course you know the phase of the outcoming pulse, but still you need this knowledge of the incoming attosecond pulse. I fully agree with you. By now we know that high harmonics are attosecond pulses or attosecond pulse trends. So
okay, great.
I think we're on the same page.
Thank you. We have also Prita D, who is asking, could you tell about the normalization procedure for the spectra against the laser fluctuations?
Yes, so maybe I switch back here. Perhaps I can do this quicker. So this one here. So we place a thin film in front of the sample, right? So like probably shown here, right? So this is this thin film. It's just a few nanometers thick, actually. So it's silicon nitrate. And what we then have is here the incoming beam, right? And the reflected and the beam that's reflected directly at this film here. This is the reference, right? So this means that for every exposure, we know the incident radiation, right? And so what's shown here in dark blue, this is the radiation that's reflected at the sample. So it has imprinted, this radiation has imprinted the fluctuations of the XUV radiation and imprinted the spectral interference from the sample, right? And because we know the incident radiation, so the incident spectrum, we can normalize this out, right? And the mass here is shown here, right? So we have the reflected light, we have the transmitted light, we have the signal. So it goes twice through the sample, so therefore it needs to therefore this square here. And in the end, so you get here this normalized spectrum.
Okay, are you fine? You can speak. If you would like, Prita, you can unmute yourself and pursue the question. But if it's fine by you, you don't need to. Are you fine?
Yes, yes, I'm fine. Yes, I got it. Yeah, thanks.
Thank you.
So
So,
perhaps, so what you also have here is the residual harmonics. So what we try to do is to get a flat spectrum, because a flat XUV spectrum, because we want to measure the modulations, the spectrum modulation produced by the sample, right? So we don't like this harmonic structure on it. Right, and we do different things in order to make the contrast of the harmonics as low as possible. But this is here the residual contrast here, and by measuring this live reference, as I just explained, you get basically a flat spectrum, right? So there are no spectral modulations here. So this is here done without a sample, right, or with a flat sample.
I see. Yeah, thank you.
so are there further questions? I mean, it's nice. We had a lot of participations today here, people asking questions. We have another one. Patrick Dupre, is terahertz reflected signal fully linear?
Sorry, once again?
Is terahertz reflected signal fully linear?
Is there a what?
Terahertz, THz.
Ah, terahertz.
That's my accent.
Well, I don't know why terahertz. What has this to do with terahertz? We are speaking about the XUV, right? But there's no non-linearity. Yeah? So the field strength is too weak in order to have non-linearities to the best of my knowledge, right?
He's asking, is the signal not terahertz? Because he's saying it's XUV, but I think there may be a misunderstanding. Would you like to unmute yourself and pursue the question? Hello, Patrick? If you wish, you can unmute yourself. We are not quite understanding what you mean. Okay, I don't know.
Mm-hmm.
Sorry, I dialed. Okay, TGZ instead of, is TGZ? Maybe it's easier that you talk, no?
TG.
Yes, I don't know. TGZ instead of the. Yeah, I don't know. Okay. Go ahead. Just a typo. Yeah, but you still have a question, right? Anyway. Okay. No worries.
Okay, good.
Good. So, do you have further questions? Does anybody have any question? Would you like to say hello? Because, I mean, if you don't want to be on YouTube, I mean, I'm okay with it. And Gerhard seems to be also okay. I just discovered today he's a fellow YouTuber. But if you would like to talk to him in a more private setting and not have your questions all over the internet, just hang in there, because we're going to finish the streaming right now. So for those who are watching, thank you so much for joining. This is, in fact, the last Atto Fridays seminar of this year. So I wish you great holidays wherever you're joining from. I thank Gerhard Paulus once more for this great talk and for
Yes, thank you for having me.
and for taking the time to talk to us.